So I'm going to tell you about this project we've been developing at the Mount Desert Island Biolab for about 8 years now. It's evolved quite a bit from initially a very concise vision of curating genes of toxicological relevance in marine organisms, because there's a marine focus to the institution where I work, and now it's become a much more broad-based effort to try to go into the literature and not forget about the legacy data and think about how that can be used to complement some of these much more high throughput approaches that we're hearing about today, and use this information in an integrated way to connect pieces between chemicals, genes, and diseases. So let's see. So when we first started this project, it was starting to evolve into more of a network-based project. We were looking to answer certain types of questions that we felt were not achievable given the existing resources at the time. So I'm going to just use a couple examples of individual chemicals or genes throughout this talk, but keep in mind that you can substitute pretty much anything, any chemical or gene of interest to you. So we were interested in things like which diseases are associated with something like bisphenol A, which genes that are affected by bisphenol A may be functioning in development, what biological functions are affected by BPA, molecular pathways, so you can kind of get the idea. If a lot of this data was at the time housed in literature that was being indexed by PubMed, and we all know if you go to PubMed and search for something like TCD, you're going to get thousands of records, and it's not really easy to sort of parse into the particular papers or the particular pieces of information that are of relevance to your particular question. So what we decided to do was to take a manual curated approach. So this is going to be a very different sort of talk than what you've heard so far. So what we do is we have a number of PhD level scientists who go into the literature, and they manually curate specific aspects of the data that we thought would be appropriate for answering some of those questions that I posed on the previous slide. So obviously this is a very simplistic view of the database. I'm just showing you sort of the major data points, so chemicals, genes, and diseases. And what we were interested in capturing, although there are obviously an endless number of types of data that you can capture that would describe these types of relationships, we focused on looking at molecular interactions between chemicals and genes. And when I say genes, I'm also talking about proteins. Gene disease and chemical disease relationships. And thank you to Richard for bringing up the idea of control vocabularies and ontologies. And all of this information is captured by our curators in a very controlled way. So we use a modified version of the MESH vocabulary, which is developed by the National Library of Medicine for our chemicals. We use Entree Gene from NCBI for our gene vocabularies, and a combination of MESH and OMIM for our disease vocabulary. And there was no actual vocabulary or ontology that would describe molecular interactions between chemicals and genes, so we developed our own. And it comprises right now about 55 different terms. So just to give you an example, this can comprise something like a chemical binding to a receptor. It can mean a chemical affects the methylation state of a particular promoter. So it can get actually very specific. Then in addition to bringing in these types of curated data, we also, I should say, as we curate our data, we prioritize it by chemical. And we have a list of chemicals that is constantly evolving based sometimes on user suggestions. We've had several collaborators approach us and say, we're interested in this particular disease or this particular chemical. Can you go out into the literature and curate that and integrate it with your resource, which we've done. We also have several of the EPA chemical lists and NTP lists that we've heard about earlier today. So right now, the state of our curated interactions, we have over 180,000 chemical gene interactions that are curated from the literature, almost 6,000 chemical disease relationships, and almost 10,000 gene disease relationships. And in addition to that, this is just our curated relationships. We also have our actual gene, chemical, and disease sets are actually quite large, and have other associated integrated data connections, but not necessarily manually curated associations. We incorporate a number of other large data sets that probably most of you are familiar with that are integrated on such a level that you can actually query across them, and I'll show you an example of that. But things like the gene ontology, which describes functions of proteins, information that come in entree gene records. The KEGG pathways are associated with genes and diseases in the database, and then of course OMIM for genetic diseases. And so by virtue of this integration, what we hope to do is provide a framework by which if we have individual relationships, such as a chemical gene relationship, and then maybe another resource or another publication has a relationship between that same gene and disease, we therefore can infer a relationship between that chemical and that disease. Now, we don't say this is a fact. What we do is we present that as an inferred relationship. Similarly, between integrated data, so I apologize, this is looking a little funny from a Mac to PC conversion, but so if we have a chemical gene relationship that we know is in the published literature, we have all of the gene ontology annotations for that gene, then we can make an association or ask the question, not only what genes may be affected by the chemical, but are there particular biological functions that are implicated by that chemical? So I'm just going to take you through, this is one of the deadly things of having a database project, that you have to show people lots of screenshots, so try to make this as painless as possible. So this is the home page. The website I'll show you later. It's ctd.mdibl.org, and it's publicly available. No passwords necessary. So you can come to the database and you'll see a navigation bar at the top by which you can access data through a number of mechanisms. There's a search page that gives you a couple of options for sort of more advanced queries. And this can get very involved based on the integrated data. So, for example, a chemical gene interaction query, this means that you would be querying our manually curated interactions, and you can combine all kinds of information to ask as broad or as specific a question as you're interested in. So you could combine a chemical term, a pathway, a gene ontology term, or alternatively, you can just use a quick keyword entry. So if we were to search for bisphenol A, what you would see is this initial page, and all of our chemical pages, gene pages, and disease pages are structured in the same way. And you have a sub-navigation bar by which you can access the particular data that's associated either by integration or curation with your particular chemical gene or disease of interest. So for bisphenol A, if you were to go to the interaction page, this takes you to the page where we have all of our curated chemical, gene, or protein interactions. And just to give you an idea what that looks like, you would see the chemical here. Our chemical vocabulary is hierarchical. In this case, bisphenol A is obviously a very specific chemical, but if you had done a query of something like arsenicals, in this list here you would see arsenicals, but you would also see all the child terms, very specific terms that fall within that group. You'd see the interacting gene, the organism in which this interaction was curated. We cover organisms that comprise vertebrates and invertebrates. And we provide you with a text-based description of the interaction that we curate. So in this case, bisphenol A increases expression of ABCC13 mRNA. So it can get very specific. And then we always provide you with the original reference so that you can go get additional information about that particular interaction. So if you were to go to this gene page, you'd just get a list of these genes. All of these pages are downloadable, so you can take these lists of genes and go do something else with them on your own. If you go to the ChemComp page, This is a new page that we've added for genes and chemicals, and the idea was, based on all of our curated data in the database, if you have a chemical of interest, you might be interested in knowing what other chemicals have similar interaction profiles. And so in this case, polychlorinated biphenyls is the highest scoring similar chemical, and you can see genistein, estradiol, things that make sense in the context of an endocrine disruptor like bisphenol A. If you go to diseases, I'm going to show you we have a couple different ways that I alluded to earlier of capturing chemical disease or gene disease relationships. They can be directly curated from the literature, or they can be inferred. And in the new release, you'll have this option of looking at just direct or just inferred. I'll show you the direct first. So what we present to you here is, again, the chemical, the disease with which we have an association. In this case, marker just means that that chemical has some role in the etiology of that disease. And we also show you genes that would be the basis of an inferred relationship between that chemical and that disease. So what that means is bisphenol A, in the absence of even having literature that supports that relationship, we would have created that inference based on the fact that BPA is associated with all of these genes, and these genes all have direct relationships with prostatic neoplasms. And so I think for my work and for others, I think this is really important, because if you go to the literature, and even in papers where BPA may be documented as having an association with prostatic neoplasms, you rarely have any information about what the underlying mechanism might be. And so I think there could be real value in leveraging some of these genes and asking, is there some value to looking at what these genes are and understanding how bisphenol A may be associated with prostatic neoplasms? And so just as a quickie experiment, I took these genes and put them into something like ingenuity pathway analysis, and actually one of the top scoring predicted networks is associated with cancer and urologic diseases. And so I think in this context, then you can start to piece together what, not only that all these genes have an association with both the chemical and the disease, but they have known relationships with each other. So moving along this navigation bar, we also include the KEGG data. So we do a similar inference where we would associate bisphenol A with acute myeloid leukemia based on the fact that BPA has associations that we've curated to all of these genes, and these genes all have an association or are members of the KEGG pathway for AML. So I'm gonna, there are actually other tabs here. We're sort of reorganizing some of this. You can always go to a reference tab. That'll show you all the references we have for that particular chemical. We also have a links tab. So we, in addition to integrating large data sets like KEGG and GO, we also link our data out to a lot of other databases, so places like PubChem, and I hope in the future some of the EPA databases we're hearing about today. So I'll just show you a couple real simple tools that we've added in the last year or so to help you ask more customized queries. So we have a batch query, which allows, rather than asking particular one chemical at a time kind of questions, you can put in a list of either chemicals, diseases, or genes of interest, and then choose the type of results that you'd like to retrieve, either curated chemical gene interactions, pathway associations, disease relationships, or GO associations. We created a Venn viewer tool, which allows you to compare associated data sets in CTD for up to 3 either chemicals, diseases, or genes. And so again, you can choose your input, you can choose the chemicals that you want to compare, and then choose the associated data sets. So just as an example, if you were interested in arsenic, which my lab is, and you know that folic acid has some effect on the toxicity of arsenic, it's not understood what the basis of that is, You might look at what the overlapping genes are between these two chemicals in terms of what they interact with. You might also look at what some of the overlapping pathways are. And so these unions may then give you some way to start focusing in on what the mechanism is between the interaction of something like folic acid and arsenic. And then finally, my gen view. This was really for me, but it's actually been helpful to a number of other groups. And so taking a list of genes of interest to you, such as a list that might come from a microarray experiment, and then comparing it to what's in the literature, essentially. And this can be really helpful for validation of what's otherwise somewhat noisy microarray data. I'll show you an example of that. So Robert also saved me the description of a zebrafish. My lab works on zebrafish, and we're interested in the developmental effects of arsenic exposure as well as dioxin. So in this particular experiment, we exposed zebrafish embryos to very low levels of arsenic, 10 or 100 parts per billion, and found 99 genes that were significantly differentially regulated. We compared those to arsenical data, curated interactions with genes in CTD, and found 20 overlapping genes. When we take those 99 genes and create a network from Ingenuity pathway analysis and compared these 20 genes to this network, we found that half of them essentially were members of this top scoring network. And this is particularly important because this network is associated with leveraging an immune response. And it's known that very low levels of arsenic exposure compromise an adult organism's ability to leverage an immune response through physiological assays, but there's never been really an explanation for what the molecular underlying components of that is. And so now we're exploring what the role of some of these genes in sort of imprinting that effect during a developmental stage. And these results were published in EHP this year. Another group, Chris Portier's group, took CTD data and integrated it with, took our chemical gene relationships and integrated it with gene disease relationships from another public database, and created these very large networks to try to understand whether chemicals are clustering significantly with specific diseases based on underlying gene interactions. And this is a really nice paper that shows that there's really some potential value to taking these data sets, integrating them, making hypotheses, and developing connections between these chemicals. In this case, these pink diamonds represent retinoids, and associating them with diseases, some of which had previously been shown to be correlated, and others there was no correlation previously made. So it begs the question that maybe there are potential associations there that should be explored further. And then finally, we started a kind of interesting project earlier this summer with Mark Corrales, who's at the EPA, who's interested in the environmental etiology of autistic disorders. And so we actually specified a couple of our curators to go out and look at autism-associated data from both the literature and from other databases. And now we have curated 213 genes with associations with autism. We took those 213 genes and took all the chemicals in CTD for which we have curated interactions. And now Mark is doing the hard work of thinking about what those chemicals mean and how you might distill that list into coming up with candidate chemicals that may help to explain the potential environmental effects on autism. So next, what we'd like to do is take a lot of our data right now is very text-based, taking this and adding visualization capabilities so that you can leverage this text-based information into images that the user can interact with on a more dynamic level. We also are looking at integration of additional data sets, so particularly SNPs that we know have associations with diseases, There are a lot of chemical codes resources at the EPA that would help us and help the user categorize chemicals that may be associated with a disease or gene of interest to them. And then I'm just going to touch on text mining a little bit. Probably the biggest limitation of curated databases is the workflow. So several databases have tried to rely exclusively on text mining. This becomes a disaster, and for a lot of groups. We've gone in and tried to analyze particular data sets to figure out in other databases that have relied on text mining and find that it's very hard for the user to figure out what is real and what is not. And so the other side of that is manual curation is very slow. And so we've started working with a group at MITRE and University of Colorado who are experts in natural language processing, and we now have a project with a paper coming out probably in the next month or two where we've done really exhaustive studies of tools that we have put into a pipeline that can go into the literature and do a quicker job, essentially, of finding candidate papers for chemicals of interest to us and use that in combination. We're not hoping to replace the manual curation, but use it in combination with the manual curation to do maybe an even faster job of pulling out the particular pieces of information that are of interest to us to increase our coverage. So that being said, the other limitation of doing this is that the data is as fast, the data is as complete as fast as we can curate it. So any mechanism by which we can increase that, the coverage, but do it in a controlled way so that it doesn't start becoming sloppy. So that's where we're trying to find that nice balance. And I'll just stop there and acknowledge there are a small but efficient group of people working on this project. It's funded by NIEHS and NCRR. And I just included my email down here. I really encourage people to contact us with any ideas, criticisms about the database. If you have particular data sets you'd like us to curate, we're happy to do that. We also have a general email that is accessible all over the database itself if you want to register any comments to us. And the other thing that we've started to do in the last 2 months is now, as we curate the literature, we send out, and maybe some of you have received these emails, we send emails to all the authors for which we have curated data, and we ask you to take a look at the database. If you have comments, if we've gotten something wrong, tell us. And it's also a help to sort of build awareness among the community that we're here, and we'd like to be as valuable a resource to you as we can, so we need your feedback.
I have a small question while George's getting to the microphone. So these databases clearly enable a lot of new things, but they're also potentially prone for misuse. So some of the misuses or unauthorized uses could be just people going in and drawing some links, and without further testing or verification can make some connections, because data is very vast and you can make a 2-degree or 3-degree or 5-degree association between anything and anything else, as we know. So do you try to think about that and put some safeguards into place? Your inferred relationships, do you assign a probability or truthness score to it? I mean, how do you deal with that?
Yes, we think about this all the time. And at the moment, the way the inferences work now is like everything's transitive, which we recognize has some issues. And we've just recently hired a biostatistician whose time and effort is going to be very much focused on our project. And we're actually, in the next probably 2 releases, we will have a way of assigning essentially a probability score to these inferences. So the inferences, it's kind of a tricky thing, because you can have an inference from a chemical to a disease based on a single gene. Well, that may not be as interesting as something that's based on 15 genes, but what if that one gene is really important? So we don't necessarily want to eliminate that, but we want to create a ranking system. So the onus is always on the user to make their choices. And anything that we infer, we say is inferred. So.
Yeah, I'd actually like to talk a lot more about that, but in the panel discussion where everybody else can talk about it. Just in terms of specific questions, my question is about one of the numbers that you put up, which is for the chemical gene interactions, you had 186,000 data entries, and maybe I'm just misunderstanding what that means. It seems to me like you could do that with 9 chemicals with a typical microarray experiment, or even fewer if you were doing chemical plus dose kind of thing. So what is the 186,000?
So those are unique interactions between a chemical and a gene or a protein, and they comprise about 8,000 different genes and about 5,000 different chemicals. So
these are only the ones that there was some sort of increase or decrease.
Well, it's not just transcriptional effects. It's binding, it's methylation, it's phosphorylation, it covers a wide gamut
Okay.
of.
All right. So, but you're relying on the original source
The
to
published
determine
literature.
whether it's okay. So it's not you're going back into, say, the raw data file and applying a consistent criterion for. Okay. Thanks.
Ben Van Houten, University of Pittsburgh. I'm really excited to see the development of the tool. It's come a long
You've
way
been
since.
around since the beginning of that,
and
where you were there.
I just wanted to know, I think the inferred relationship, to come back to Yvonne's point, is really important to try to develop a score. But I know that's a tough problem. What organisms are you currently curating the database for? Do you include C. elegans, yeast,
Yes.
or Drosophila?
Yes. So it's vertebrates and invertebrates. And the majority of the data tends to fall into mammalian organisms just because that's where the majority of the research is. But there's certainly zebrafish and there's C. elegans, and it tends to be very topic specific though. So.
But did you include yeast? I'm sorry.
No.
Okay.
One final point, a slightly different question, although I'm interested in the p-value discussion as well later. Databases like this, historically, again, having been around a long time, in an academic setting, have a tough time getting funded over time. This is exciting now. 6 years from now, who knows? But it's just the nature of the beast. My question is, have you been working with somebody like the National Library of Medicine, thinking about the translation of this from a research effort that's funded in an extramural capacity to an effort that's funded by the government and taken over, and you focus on some more research aspects of it rather than just the day-to-day grind of keeping up with this thing?
Right.
Have you worked on that?
Yeah, I actually realized that I have been remiss that the major grant that funds CTD is an NIEHS NLM joint. It was originally sent to NIEHS, and then NLM was very generous to pitch in because we're funded by an R01 mechanism just by virtue of the fact that there's really not an appropriate mechanism for our subject matter. And getting funding has been challenging. So yes, we are interested in establishing potentially some sort of more stable, less R01 structured funding. Remains to be seen how that works out.
So, excuse me, one of the problems with manual curation is of course, that there's so much out there. And so you're talking about thousands of chemicals and thousands of interactions. How do you prioritize what to include and what not to include?
Yeah, so I showed on one of the earlier slides that we have a number of EPA chemical lists, a number of other regulatory agency lists, and we do a lot of comparisons. We prioritize ones that obviously cut across multiple lists. We have it's a hard question to answer because we can't do them all, but we try to.
Not just chemicals, what papers you actually.
Oh yeah, so we do have a list. So what we do is we prioritize it by a number of different criteria. So we have a reasonably large but small in the context of all possible published or manuscripts, journals. So we have a list of journals that we prioritize. that are tox related or high profile related to sort of help contain the landscape. We also, if it really depends on the chemical. If you go into PubMed, you may find and do a broad-based search without restrictions, you might find 10 papers or you might find 10,000. And so we start restricting it by year. We focus on more recent things in a lot of cases for chemicals that have a huge number of papers associated with them. There is some subjectivity on the part of the curators. If, they go through and they're curating the same interactions over and over again, they sometimes make a call about moving on to other types of interactions. So I think one of the things that text mining will also help us with in that sense is the ability not only to go in and prioritize the most relevant papers, but we can also say we have these types of interactions and go into the literature and say, are there other interactions that are relevant to this chemical or disease that are not currently represented in here, as a means of making sure that we've covered that scope a little bit better.
I wanted to continue a question that was asked earlier about what do you do with conflicting data, and I guess the bigger question I have is how do the curators, or do the curators make any judgment based on quality of the study or strength of the evidence? And so, for example, you've got mRNA microarray data versus something that might actually follow through with looking at some protein or functional effect. When they get into your database, are they all equal in terms of how the curator enters that data? Likewise with disease, is that epidemiology data? So how does one distinguish between, say, a case study?
Right.
Will your student who puts that at a same level as something that's like a case control study? How do you manage that?
Right. So let me see if I remember all the questions. So our curators don't make judgments about the data. The data is peer reviewed. We let the journals do sort of that triage process. So we capture what there's evidence for in the paper. As far as conflicting data, we capture it, because usually there's some underlying reason for that, right? Maybe it's a different developmental time point. Maybe it's a different organism. And that's actually, I think, another strong point of this database is that, you may see a chemical gene interaction that, say this chemical upregulates expression of a particular gene in mouse, but it doesn't do that in clam or fish. And there may be some difference in the way those genes are functioning that could help explain that. So I think conflicting data is actually really important. As far as making sort of preferred marks on data based on how it's gathered, we don't at this point. We do always provide the reference, so a user can always go to the literature, and if they go to it and they say, oh, it's this kind of study, I don't care about that, well then, okay, that's their choice. But
I'm sorry, do you capture something like dose?
We do not.
That was my next question.
Yeah.
It seems that would be another layer on top of this.
Yeah.
Is the dose, and how much focus do you put on the range
Right.
of the
So
concentration?
dose was, when we first started this project, we had all kinds of data on the table that, is curatable in a sense, but dose, knowing how important it is, we had to let that one go, because it would not be practical for us to curate that across all the papers. The way dose is captured in different organisms is so different that we let that go. And I still feel this every once in a while, but that's the way it is at the moment.
Well, thank you so much.
Thank